Cell division. Part 1. In the earlier classes, you have studied about cells. A cell is the structural and functional unit of life. New cells arise from the pre-existing cells by the process of cell division. Cell division is an important phenomenon that occurs in all living organisms. In unicellular organisms, cell division directly produces two individuals. Thus, it is a means of reproduction. In multicellular organisms, there are two types of cells. The somatic cells, or the body cells, which form the body of the organisms, and the reproductive cells, or gamete-producing cells. In multicellular organisms, life begins from a single cell, the zygote, which divides and redivides into a number of cells to form a complete organism. Now, the question arises, why do cells divide? The cells divide to produce new cells. The new cells are produced for growth, replacement of dead cells, repair of tissues, reproduction. Types of cell division. Two types of cell division occur in organisms: mitosis and meiosis. Mitosis occurs in somatic cells as well as reproductive cells, such as in spermatogonia and oogonia, before entering meiotic cycle, leading to growth and development. Whereas meiosis occurs in reproductive cells, leading to gamete formation. Karyokinesis and cytokinesis. The cell division includes two events: nuclear division or karyokinesis, leading to division of parent nucleus into daughter nuclei, followed by the division of cytoplasm or cytokinesis, leading to the division of the parent cell into daughter cells. Let us study about these processes in detail. Cell division. Part 2. Mitosis. Greek, mitos, thread. Mitosis is an equational division in which one parent cell divides to form two daughter cells. The daughter cells formed are identical to each other and also to the parent cell in every respect. In mitosis, the same normal chromosome number of the parent cell is maintained each division of the cell, and hence it is referred as equational division. Interphase. Interphase is the growth period between two successive divisions of a cell. Thus, it is a phase just before the cell starts dividing. This stage is said to be a resting phase because no external change in chromosomes is visible. However, the cell is metabolically most active and prepares itself for division. The following events take place during interphase. 1. Synthesis of RNA and proteins required for normal life of the cell, that is, energy production, growth, repair, etc. occurs. Two. Synthesis of new genetic material required for division occurs, that is, DNA molecules duplicate. 3. Volume of cytoplasm and nucleus increases. 4. Chromosomes are not yet distinguished. Phases of mitosis. Mitosis occurs in 4 phases, namely 1. Prophase, 2. Metaphase, 3. Anaphase, 4. Telophase. The various phases of mitosis are nearly similar in both animal and plant cells. For better understanding, the meiosis in animal cells is described here, and wherever variation occurs, the mitosis in plant cell is also described. Let us study about the phases in detail. Cell division. Part 3. 1. Prophase. Greek, pro, before, phases, stage. This is the first and the longest phase of mitosis. The chromatin material undergoes condensation, becomes short and thick, and changes into thread-like structures called chromosomes. Each chromosome has 2 thread-like structures called sister chromatids, which lie close to each other and remain attached at a point called centromere. As prophase advances, the chromosomes become shorter and condensed or thickened. In animal cells, the centrosome initiates and regulates the cell division. The centrosome splits into 2 small round bodies called centrioles. The 2 centrioles of the centrosome migrate to the opposite sides or poles of the cell. Soon, 2 star-like structures with radiating fibers known as asters are formed around the centriole at each pole. Between the separating centrioles, spindle fibers are formed by the aster. Towards the end of the prophase, the nucleolus and the nuclear membrane disappear. Another phase is metaphase. Greek, meta, between, phases, stage. Chromosomes become still shorter and thicker due to condensation. The chromosomes arrange themselves on the equatorial plane in such a way that their centromeres lie on the equator and the arms face towards the poles. Centromere of each chromosome is attached by a spindle fiber. Another phase is anaphase. This is the shortest phase of mitosis. The centromere of each chromosome divides into two halves, sister chromatids, so that each chromatid has its own centromere. The sister chromatids separate and begin to move towards the opposite poles due to the contraction of the spindle fibers and due to the repelling force developed between them. Depending on the position of the centromere, the chromosomes appear as U, V, or J-shaped. Ends when all the chromatids, now behaving like chromosomes, reach the opposite poles. Another phase is telophase. This is the last phase in karyokinesis or nuclear division. The events of prophase occur in reverse sequence during telophase. The chromatids or daughter chromosomes uncoil, elongate, and change into a network of chromatin threads. The nuclear membrane reappears around the chromatin network at each pole. Nucleolus reappears in each daughter nucleus and spindle fibers disappear. In animal cells, centrosome organizes itself above the nucleus, thus making the completion of karyokinesis. The karyokinesis, or mitosis proper, is followed by the division of the cytoplasm, known as cytokinesis. Cytokinesis, Greek, cyto, cell, kinesis, movement. It is the division of cytoplasm to form two daughter cells. It begins during late anaphase and is completed soon after telophase. It is different in animal and plant cells. Cell division. Part 4. How is cytokinesis different in animal and plant cells? In animal cells, a constriction or furrow appears in the plasma membrane. This constriction deepens by the end of the telophase, finally completing the division of cytoplasm. In plant cells, a cell plate is formed in the center of the cell at the end of the anaphase. The cell plate extends on either side until it completely divides the cell into two daughter cells. In animal cells, cytokinesis starts from the periphery and proceeds towards the center. But in plant cells, cytokinesis starts from the center due to cell plate formation and extends towards the periphery. In plant cells, no centrosome or centriole is there, and asters are not formed. Spindle formation still occurs and are formed by microtubules or cytoplasmic strands. Let us now study about the significance of mitosis. Mitosis maintains the same number of chromosomes in all the cells of an individual. In other words, mitosis is an equational division in which two daughter cells produced are identical to each other and even to their parent cell. This type of cell division usually takes place in the somatic cell, such as tips of roots, stem, etc. It plays a significant role in wound healing, replacement of cells lost during wear and tear. It is responsible for the growth of an organism. A fertilized cell develops into an embryo and finally into an adult as a result of mitotic cell division. Mitosis helps the cell maintain the proper size. It is a method of asexual reproduction in unicellular organisms. If mitotic cell division becomes uncontrolled, it may cause tumors or cancerous growth. Cycle Part 1. In this module, let us learn about the cell cycle. Till now you have studied about mitosis. But mitosis is only one phase of the cell cycle. A cell cycle starts from the time a new cell is formed, and it ends when it completes its own division. Then the cell cycle starts again for each new daughter cell is formed. A cell cycle may be defined as the events in a cell by which there is an increase in the mass and cytoplasmic components of the cell, duplication of DNA, and then division of nucleus and cytoplasm of the cell. Thus, a cell cycle extends from the time a cell is formed till the time it completes division. Interphase. The cell cycle starts with the interphase, during which a cell prepares itself for cell division. The interphase is the longest phase of cell cycle. It is metabolically the most active phase of the cell cycle. It has three subphases. G1 or first growth phase. This is the first gap or interval phase of cell growth and functioning before DNA replication. During this phase, 1. RNA and proteins are synthesized and volume of cytoplasm increases to almost double. 2. Mitochondria in plant and animal cells and chloroplasts in plant cells divide. 3. Now the cell either enters the resting phase, R phase, or a synthetic phase, S phase. Second phase is S or synthesis phase. This is the phase of DNA replication. The DNA is synthesized and chromosomes are duplicated during this phase. Third phase is G2 or second growth phase. It is the second gap phase after DNA replication. It is a shorter phase in which RNA and proteins necessary for cell division continue to be synthesized. Now the cell becomes ready for next cell division, that is, mitosis. The cell cycle, part 2. Mitosis. It has nuclear division, prophase, metaphase, anaphase, and telophase, usually followed by cytoplasmic division. The various phases of cell. The cell cycle does not go endlessly. It stops permanently at some point of time. Its duration differs from one cell type to another cell type. For example, nerve cells or neurons in our brain, once formed in embryo, do not divide further. All blood cells form and replace the worn-out cells at an average rate of 2 to 3 million each second. Surface skin cells are continuously replaced by underlying cells. Liver cells divide once every 1 to 2 years to replace damaged cells. Meiosis, GK, Greek, myomum, to diminish. Meiosis takes place in the reproductive cells that produce gametes. Meiosis is a modified mitosis in which chromosomes divide once and the nucleus divides twice, after which the number of chromosomes are reduced to half. Thus, the 4 cells resulting from a meiotic division have a haploid number of chromosomes. It means that the number of chromosomes becomes half in each sex cell. This is because the male and female gametes fuse during fertilization. The diploid or double number of chromosome pairs are re-established. Meiosis is a reductional division. Stages of meiosis. Meiosis includes 2 nuclear divisions. First meiotic division, reduction division, and second meiotic division, mitotic division or equational division. Thus, in meiotic cell division, all the stages, that is, prophase, metaphase, anaphase, and telophase, are repeated twice. Part 3. First meiotic division. In this division, homologous chromosomes come together or associate and subsequently segregate into daughter cells. Thus, the number of chromosomes are reduced from diploid or double to the haploid or single state. That is why it is known as reduction division. The following events take place during this division. Chromosomes pair and separate. The chromatin network undergoes condensation and shortening and changes into chromosomes. The homologous chromosomes, one received from father and one received from mother attract each other and come to lie in pairs. The pairing of homologous chromosomes is known as synapsis, and the pair is known as bivalent. Chiasma formation. Chromosomes continue to shorten and thicken. Each chromosome splits lengthwise into two chromatids, so that each homologous pair now has 4 chromatids and is termed as tetrad. The non-sister chromatids of a tetrad break open and rejoin each other. This is known as crossing over or chiasmata, singular chiasma formation. Exchange of some genes or portions of chromatids takes place between two non-sister chromatids of homologous chromosomes during this stage. Due to crossing over of homologous chromosomes, chromosomes separate out. Nucleolus and nuclear membrane disappear. The members of homologous chromosomes completely separate from each other and move towards the opposite poles. Nuclear membrane reappears, leading to the formation of 2 daughter nuclei. It is important to note that each daughter nucleus, thus formed at the end of meiosis 1, has half the number of chromosomes as compared to the parent cell. Second meiotic division. It is similar to mitosis. During this, the 2 chromatids of each chromosome separate and move to the opposite poles. Nuclear membrane reappears and 4 cells are formed. Finally, each cell formed has haploid or N, half the number of chromosomes of the original cell, diploid 2N. Significance of meiosis. 1. The number of chromosomes are reduced to half in the daughter cells. 2. It results in the formation of haploid sex cells, sperms and ova, which after fertilization restore the diploid number of chromosomes in the zygote. 3. During crossing over, which occurs in meiosis, part of chromatids are exchanged between homologous chromosomes, which bring about variations in the offsprings. 4. The 4 chromatids of a homologous pair of chromosomes are passed on to 4 different daughter cells. This also causes gametic variation. 5. It avoids the multiplication of chromosomes and thus maintains the stability of species.